In this video, we're going to talk about the second step of photosynthesis, and that's the Calvin cycle. So here's Melvin Calvin. He's the guy that the Calvin cycle is named after, and this is basically the process where we take carbon dioxide and fix it into organic molecules, such as sugars. So let's look at that in a little bit more detail and review the light reaction. So remember, in the light reaction, we take light energy and we use that light energy to excite electrons, and those excited electrons are then passed down an electron transport chain where they make ATP, again the energy of the cell, and they also then are excited again in a second step, and that those excited electrons are then used to reduce NADP into NADPH, again a high energy reduced molecule. So the ATP and NADPH then from the thylakoid membranes are going to be passed out here into the stroma, the area outside of these thylakoid membranes, where they are going to be used to power the fixation of carbon, where we take carbon dioxide and fix it into molecules. So let's look at that in a little bit more detail. Okay. So here's the Calvin cycle, but you don't need to worry about all these intermediates here. So this is more information than you need. Just kind of look at what's going in. We have ATP, which is, again, providing the energy to make these high energy reduced bonds, right? We're making now high energy carbon hydrogen bonds, and that takes energy. And we're also oxidizing NADP and using that oxidation to reduce further the molecules in the building of the sugar, right? So we're taking 3 carbon dioxides per cycle here, and this is going to go around a couple times to complete it. But each time we build this molecule called G3P. That's a 3 carbon molecule that can then be assembled into 2 of these to make a glucose, or we can also produce amino acids, fatty acids, or other organic molecules from this main building block, this 3 carbon sugar. Okay, so basically we have ATP providing the energy to reduce and make high energy organic molecules, and also NADPH providing, again, through its oxidation and providing hydrogens and their high energy electrons to further make this high energy molecule. So again, here's the cycle in sort of photosynthesis in the big picture. So we have the light reaction over here with the thylakoid membranes producing ATP and NADPH, and then over here carbon fixation occurring, and this G3P, this sort of first product, can be used to make sugars or starches, fatty acids, amino acids, etc. But we have a problem when we have photosynthesis going on, because we have oxygen being produced in the same place we're trying to build these high energy reduced molecules, right? So we know that oxygen wants to oxidize. It wants to rip off those high energy electrons and form low energy products. And it will actually bind to some of these intermediaries and disrupt this process in a process called photorespiration. So we sort of have this problem. We need to not have the oxygen around while we're doing the Calvin cycle. And in a chloroplast, they're right next to each other, so it's hard to not have oxygen there. So obviously you want to keep your stomata open during the day to get rid of all that oxygen and reduce this oxidative process, this photorespiration. But if you're in a hot climate, leaving your stomata open all day means you're going to lose a lot of water as well as getting rid of the oxygen. So there's this kind of this conundrum, this problem with needing to get oxygen out but also not wanting to lose too much water. So now we're going to talk about 2 adaptations that plants in hot climates have to avoid this process or this problem. So first off, corn, which is again one of the universal staples grown in a lot of hot, dry, or hot climates, has this adaptation whereby, again, here are the mesophyll cells. So these are the main photosynthetic cells, and they're producing a lot of oxygen, and that oxygen is going to be diffusing, hopefully, out through the stomata out here. Okay, but again, you don't want to do the Calvin cycle in there with all that oxygen if you're going to it's going to interfere with this process called photorespiration. So what they'll do is they'll take the carbon dioxide and they'll shuttle it down here into one of these bundle sheath cells. They're basically the cells that protect the vascular tissue where the sugars ultimately get sent to different parts of the plant. Okay, so by taking the carbon and fixing it into this 4 carbon molecule and then sending it to these bundle sheath cells, we're now separating the Calvin cycle from the light reaction. So the oxygen is being produced up here and it can diffuse out of these mesophyll cells, and then the Calvin cycle can occur in the bundle sheath where there's less oxygen to interfere with the building of these organic molecules. So that's C4. Again, we see that in many tropical grasses and in corn, and it allows them to grow in hot climates without losing too much water, without having to leave their stomata open to the point where the plants die of water loss. Other plants, instead of physically moving the carbon to a different part of the plant tissue, they'll actually just switch off and on when they open their stomata. So this is called CAM photosynthesis. We see this in pineapples and other tropical plants. What they do is open stomata at night, take carbon dioxide in. Now you can't have carbon fixation occurring because you need light for the light reaction to make the sugars. So they're going to take that carbon dioxide and they're going to convert it into an organic acid called malic acid, and they're going to store it in the vacuoles within the cells. Basically what they're doing is they're filling up this big tank with stored carbon at night. Again, opening the stomata, and at night it's cooler, so they're not losing as much water. Okay. Then during the day it's getting really hot, but they have the light they need for the light reaction. They're going to close the stomata, so they're not going to be able to get the carbon dioxide, but they already have it stored up in their vacuoles. So now they can convert that back into carbon dioxide and utilize the Calvin cycle in the day with their stomata closed. Okay, so they don't need to deal with this problem of leaving stomata open and having, carbon dioxide in and oxygen out. They can basically allow for the carbon dioxide in at night and then store it up and then do carbon fixation during the day with their stomata closed. And again, avoiding dehydration and excess water loss in really hot climates. So these are two adaptations, right? So in C4, what we see is we're physically moving the carbon dioxide in terms of a 4-carbon molecule and doing the Calvin cycle in a separate location away from the light reaction. And in the CAM process, we're actually doing it at a different time. So we're taking the carbon in at night, storing it in this organic acid, and then doing carbon fixation during the day without having to open the stomata and having to deal with the excess water loss. So these are just two adaptations that plants use to avoid water loss and allow photosynthesis to occur more efficiently in very hot climates. Okay, and that's it.